another DeSci party. We are here today with Clarice Aiello from UCLA. She is a professor of quantum biology, a quantum engineer, actually, and we're going to be diving into what that means at the biological level, at the quantum level, physics level, and then also some of Clarice's experience working in participating in decentralized science. So without ado, I never like to take away the opportunity to provide your own introduction. I know you've definitely been experienced. I checked out some of the other podcasts and things you've done. So thanks again for joining us today, and super excited to have you here. So Clarice, please provide a little introduction, your background, and
Awesome.
and how you got to what you're doing today.
So, Elijah, thank you so much for having me here. I am super happy to be talking to all different sorts of audiences about what we mean by quantum biology, what we want to do with it. And, well, first of all, I think you need to know where this accent is coming from. I was born and raised in Brazil, so that's where
Mm-hmm.
my accent comes from. And yeah, I am trained as a quantum engineer. So this means that I build instruments to study and control things that are so small and so well protected from their environment, they're better described by the laws of quantum mechanics, as opposed to the laws of classical mechanics that rule everything big around us and that we usually learn, say, in high school, right? So it turns out that one can actually prove that if you use a tiny quantum object, like an atom, an electron, or a molecule, if you use a tiny quantum object as a sensor, your measurement is improved. In other words, the sensor quantumness enhances the measurement. If you use this quantum object as a sensor, you can
Mm-hmm.
actually benefit from that. And indeed, people have been developing technological quantum sensors for more than a decade now, and many technological quantum sensors are already put to use, for example, to sense temperature, say inside cells, to look for oil. There is a company, a well-established oil company that actually prospects for oil using a technological quantum sensor. But that's not what I'm here to talk about. I'm here to talk about the fact that there is incomplete but compelling evidence that nature might be using some of the same tricks uh, than uh, than uh, than the tricks that are used by technological quantum sensors in order to work and in order to work optimally. So what I do in my lab at UCLA, which is the Quantum Biology Tech or QuBiT lab, is to study and try eventually to control those nature-made quantum sensors. Okay, and I can explain a little bit later why we do this, and or shall I just start explaining what we're doing? Or
Yeah, no, take it away because I think that's an interesting start. I'm curious about what these potential impacts are in nature and what these biosensors are. But no, please continue.
yeah, so again, there are many ways in which nature is exploring quantum mechanics to function and to function optimally, right? So, if you will, there are many flavors of this emerging field called quantum biology that wants to explore those endogenous quantum degrees of freedom explored by nature. For example, there's evidence that um, photosynthesis uses quantum mechanics to function very well, and that's the reason why plants are way more efficient at gathering photons than humankind made solar cells, for example.
Mm-hmm.
There is evidence that enzymatic functioning, like that your enzymes that catalyze all the reactions in, well, many chemical reactions in biology, that this catalytic function is mediated by, for example, a quantum phenomena called tunneling, electron and proton tunneling. Okay, so your
Mm-hmm.
enzymes, reactions can only be catalyzed because quantum mechanics seem to be at play, all at room temperature. In my lab, we study a flavor of quantum biology that has to do with spins. So, and just before we started, I was telling Elijah that I always travel with my spins. I never leave home without my spins. So, um, spins are a merely quantum property of matter. For example, electrons have spins, atomic nuclei have spins, and spin is as fundamental as, say, charge and mass. And in the same way that, say, mass tells us how an object interacts with the gravitational field, charge tells us how a object interacts with an electric field, spins measure how an object interacts with magnetic fields. And if you think about it, when we say, positive charge or negative charge, say an electron is negatively charged, there are some ions that are positively charged. This only means that those two, like positive charges and negative charges, they interact with electric fields in a different way. For spins, we don't call them like positive or negative spins. We usually use an arrow and we say it's like up spin or down spin, spin up or spin down. And spin up or spin down just represent two different ways of a quantum object interacting differently with a magnetic field. And it turns out that it's been known from basic chemistry, from like test tube chemistry for decades, that there is a class of chemical reactions that depend on spin. Here's how this works. There's a chemical reaction that is happening, and at some point, the chemical reaction comes to a crossroads, and the chemical reaction effectively looks for a certain spin state of a electron, a particular electron. If this spin of the electron is up, the chemical reaction continues through one branch. If the spin of the electron is down, the chemical reaction continues through another branch. Importantly, the final products of those two branches are macroscopically different. So a finicky quantum property of the electron spin can actually macroscopically alter the final products of a chemical reaction. Moreover, if at the point when this chemical reaction comes to this crossroads, this electron interacts with an external magnetic field, for reasons that I won't be able to prove here, this is a bona fide quantum sensing process where this spin is sensing this external magnetic field, and it turns out that this external magnetic field might actually alter the spin to be more likely to be up or more likely to be down. So it's in this way that a magnetic field can alter also the final product of a certain class of chemical reactions, and at much longer time scales. This interaction is very, very brief, but everything happens downstream. So in test tube, there is no doubt that there is a quantum sensing phenomenon happening that alters chemical reactions. And there's also evidence, macroscopic evidence, with whole organisms or with cell cultures. There's evidence that they are reacting to magnetic fields in a way that is consistent with their behavior, their physiology, being driven under the hood by a spin-dependent chemical reaction. So what we want is to learn how to deterministically change those spin-dependent chemical reactions in order to drive physiological outcomes. Let me give you some examples of chemical reactions that depend on spin, at least in test tube chemistry, and for which there's correlational macroscopic evidence. For example, and this is the poster child of this flavor of quantum biology, birds. Birds, when they migrate, it's known that they use the tiny magnetic field of the Earth as a compass, at least as a partial cue. And the magnetic field of the Earth is weaker by orders of magnitude than the magnetic field that you sense when you bring your cell phone close to your head. Wow. So how might they be doing this? The most strongly standing theory is that they do so mediated by this type of spin-dependent chemical reaction. Also, the production of reactive oxygen species that are signaling agencies or that can cause harm if you have too much of them. There's evidence that the production of reactive oxygen species is dependent on magnetic fields in a way that is consistent with them being in a spin-dependent way. Also, the regulation of cellular respiration rates, acidification rates, stem cell growth, yield of DNA repair. Those are just some processes that seem to depend on magnetic fields in a way consistent with this spin model. And I think that, and I'm almost at the end of my initial spiel here. It is well understood, at least in theory, and for test tube chemistry, that those chemical reactions that depend on spin, they depend on relatively weak magnetic fields for reasons that are well understood. This means that if you put proteins that can sustain spin-dependent chemical reactions inside a big magnetic resonance machine, the 5 Tesla magnet is not going to influence the chemical reaction to go more through one branch or through the other branch. The magnetic fields that make a big difference are relatively weak magnetic fields, such as the Earth, such as the magnetic fields that you can easily produce with electronic devices, right? So, for example, if you grow cells or if you grow embryos inside chambers that shield very well the magnetic field of the Earth, which is tiny, you can induce like macroscopically observable embryogenesis errors. Okay, so there's an experiment where tadpoles were grown in a very controlled way for 2 days in the absence of the Earth's magnetic field, which is already tiny, right? And people have seen like that embryos were malformed, were not viable at about a rate of like 30%. So we're not even putting a magnetic field, we're taking the tiny magnetic field of the Earth out and we're creating all sorts of like weird stuff, which brings us to which opens up this field from like space exploration. Like if you want to colonize Mars, what's the magnetic field on Mars? Can we reproduce in Mars? Can we grow lettuce in Mars? To more pedestrian things such as, can we learn how to deterministically tune those endogenous spins, right, in a way that we can drive physiology, say, in order to therapeutically change something, right? So there's also evidence that weak magnetic fields, they regulate in some weird way, but still consistent with the spin model, they regulate ion channel functioning. So here's potentially, maybe in the future, another way of doing optogenetics. Nowadays, when people want to control, the opening and closing of ion channels, they genetically encode a protein and they shine a laser. This laser opens and closes ion channel. Well, here we don't need a laser. Everything is genetically encoded. Magnetic fields can penetrate very deep. So is there a way of doing this quantum genetics or quantum manipulation of ion channels? Again, right now, this is absolutely science fiction, but my take is that we need to start doing this type of research now so that we can benefit from that, say, in 25 years. And this is the end of the spiel. Oof.
Okay. That was a good spiel. You really can sum it up well, because there is so much to cover. But it's to me, what kept coming up is right now, I think it sounds like we're at the place with quantum biology where, we're looking at these things like necessity and sufficiency and correlation causation, right? Is this participation, is this being placed in the magnetic field sufficient to cause a change, right? Or not being in the magnetic field? And that's sort of a lot of where we need to go for it, right? And we'll figure out the mechanisms as we find maybe these examples of a protein that can be magnetically controlled, and how is that dependent on the spin state and
exactly.
So on, right?
And like the data that exists, they're correlational, right? So and they go from test tube chemistry to like macroscopic things that react in a way that is consistent with a macroscopic model. But it's very hard to as you say the bird is doing its thing, because there's a spin-dependent chemical reaction happening somewhere in the bird, right? So this is why in my group, we want to take a bottom-up approach, and we want to start, say, dealing with single cells. We want to go one step further from test tube chemistry and start doing quantum-inspired experiments inside, say, single cells, right? To in the same experiment look at the quantum mechanical degrees of freedom, like the spin, like measure the spins and see how a certain spin state affects downstream functioning in a very controlled way. And this flavor of, and quantum biology in general is sort of a little bit plagued, because currently there's a lot of theoretical quantum physicists doing theory, but most experiments are done by biologists and chemists. There's no problem. Those experiments are beautiful.
Mhm.
They are necessary, but again, they're not sufficient. What we need, in my mind, is to bring technologies developed for mainstream quantum experiments and start applying those technologies onto biology, treating quantum degrees of freedom in biology as if they were, because they are, bona fide quantum objects, right? And I think that what we need right now is for quantum engineers to be involved, and especially to start, stepping up the instrumentation that we can use to probe unambiguously, right, with a cause and effect relationship. Uh, if nature is really using quantum mechanics to function, I can say with certainty that right now there is zero experiment on Earth that can unambiguously show whether nature is using quantum mechanics to function. Okay, everything happens either for proteins in solution or for birds or for
Mhm.
flies that
Right.
respond in a way that is consistent. So this is why I think we need those very, if you will, high-tech experiments that we're developing in our lab.
Yeah. Yeah. That sounds like, yeah, it must be difficult to run an experiment with a bird that has a magnetic, I don't know, thing, cage or jacket or whatever.
Yeah,
I could
exactly.
imagine.
Those experiments, they are extremely
Yeah.
hard, and
Yeah.
people get 20 birds during migration season. They put them in
Yeah.
cages, and they want to
Oh, no.
see
Yeah.
which way the bird goes out, and they mess up with the magnetic fields, and the birds want to go to another way. Again,
And those poor
this
birds.
provides
Yeah.
this provides beautiful correlational data, right? This is
Right.
necessary, but it's not
Right.
sufficient. We do
No.
need to start putting high tech into quantum biology, high
Right.
quantum
Right.
tech.
Yeah. So is that what you're doing at QuBiT? Is that what's going on in the lab? And
That's
and maybe describe a little bit
that's
more the type of
exactly
experiments you're doing?
what we're doing. We are building glorified microscopes with coils, but our microscopes are such that they are inspired by the field of quantum sensing. Actually, we are gearing up to be able to measure the state of the spins in the same way that one would be able to measure the states of the spins in a technological quantum sensor. So
Mm-hmm.
it's like microscopes that are going to both look at the quantumness of 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 of, of the proteins
Mm-hmm.
and downstream physiological processes. Yes.
Okay. Cool. Yeah. So you have a multimodal readout, and it's able to also influence, right? Same. Yeah.
Yes, and I can actually tell you exactly how it works. Our readout is by fluorescence.
Mm-hmm.
So, for example, there is a very famous technological quantum sensor, which is basically a electron spin in diamond, in the material diamond. And the way that people read out the quantum state of that sensor, that also works at room temperature, by the way, it's called nitrogen vacancy sensor. Some of you of the audience might have heard about this sensor. It's a very standard sensor. It has the very interesting property that it emits light, it fluoresces at different intensities if that particular sensing electron spin is up or down. Okay, so just by looking at how strongly that technological quantum sensor is emitting light, you can actually infer the state of the spin, up or down. It turns out that for very similar reasons, the proteins that can sustain such spin-dependent chemical reactions, those are most times fluorescent proteins that have a chromophore, and they also happen to fluoresce at different intensities if that particular electron spin that you care about is up or down. So in the same way that people look at fluorescence intensity emitted by the technological quantum sensor to read out if it's up or down, we're also going to be looking at the fluorescence emitted by the proteins that can sustain such spin-dependent chemical reactions to read out if the spin is up or down. And then what we want to do is, given if the spin is up or down, or messing up with the spin state with the magnetic fields, read out via standard fluorescent biomarkers, right? Say reactive oxygen species markers, redox markers. What is the consequence of a certain spin being in a certain state in the cell? downstream at much longer timescales.
Mhm.
This is
Right.
like the working principle of all our experiments, basically.
Yeah, and that's really the trick too. I think and that's the leap people have to make cognitively when they start thinking, okay, what is quantum biology even, right? We're going all the way down from these big macro right organism level,
Yeah.
behavior level, right, things that we're observing all the way to the level of spin and properties of the quantum nature. So super cool. The ability to bridge that with a single experiment. I think that might have been something you mentioned in a previous podcast. Is that it was at the length scale, or is it? I guess now it's also the time scale too, right?
Exactly. We want to bridge those two length scales and time scales, right?
Yeah.
Because
Yeah.
The quantum processes in chemistry, in test tube chemistry, they occur very fast, right?
Right.
And the physiological processes, they occur at very large length scales and downstream, like at much longer time scales. So we want to bridge those two length and time scales, exactly.
Yeah, super cool. And so it's going well, I assume. The microscope is being made. You guys got the experiment. It sounds like you have the protein that at least you can do the readout. That alone is a huge tool. And demonstrate
and yes, and it doesn't mean. I mean, we are going. I have a superb team that mostly comprised of experimental quantum physicists. I think we're really the first quantum bio group that is composed primarily of nanoscientists and experimental quantum people, which is, I think, what we can contribute to. None of us, I think, has a lot of chemistry or bio training, but I think we can contribute with the instrumentation development. But and I think that's a good hook to talk about DeSci, right? Quantum biology is a aging field in the sense that there is zero incentive for young blood for early career scientists to enter this field. Most people pivot to quantum bio in the second half, in the last third of their career, because there is no steady funding for that, right? People are more established. When people have a source of funding that they can rely on, they pivot to doing such experiments. There are no standing calls for quantum bio at NSF. NIH folks have already told me in those words, like about a year ago, like, Clarice, forget about it. The NIH is not ready for quantum bio. There are foundations that give a patch of money here, a patch of money there, but that's no healthy way of building a career. So there's still a lot of stigma related to quantum biology. The bio people, it's my task to share why I think looking at quantum degrees of freedom in biology is beneficial, and they don't have the training necessary. So it's part of my job to educate them in the best way possible, right? And to learn from them. And the quantum people, the mainstream quantum people, are very hard to deal with because usually quantum things happen at very low temperatures under very controlled environments. And even though there are examples of quantum objects that live at room temperature and under messy environments, such as the technological quantum sensor that I just described, sometimes the mainstream quantum people won't engage. They're very dogmatic in their ways, and they refuse to engage. So that's very hard. So we are really in no man's land, which makes it exciting because we're learning things all the time. But funding wise, it's very, very hard. People don't really understand what we want to do. And most of quantum biology, about, say, at least 80% of quantum biology, is some version of electromagnetics in biology. For example, if there's any physicist in the audience listening to this podcast, I like to describe what we're trying to do as light matter interactions where matter is biomatter. This is a sentence that a physicist should be able to understand. And electromagnetics in biology is something that carries like this weird baggage. Research for electromagnetics in biology has never stopped in the East, in Russia, in Japan, in China. But in the West, it's still taboo. People think, oh, it's like some new agey woo-woo weird effect, and it's not. The data that exists, there has been there's more than 40 years of data showing that electromagnetics in biology is a thing, that electromagnetic fields are altering physiology in a way that is not very systematized and whose mechanisms are not very well understood. But there is a big wall there that people sometimes don't even want to hear about this. And this has to change. And I think that, if I could have it my way, I think that quantum biology is a huge opportunity for decentralized science. And one of the things that I want to do with my research and with my academic life or my professional life is help bring this field to the mainstream, if you will, electromagnetics in biology, or the next level, like quantum biology.
Yeah. Right. And that's so true. There's the decentralized science movement, right, is very much a big part of it is getting funding and access to funding and also this kind of getting past this bias is essential censorship in the funding because of the funding, exactly. to these different things. So what's been your experience working with DeSci projects, getting involved in decentralized science? We actually met a couple months ago back at the Gitcoin this event about Gitcoin, and you were presenting there. So how did you get involved? How did you hear about it? And what's your experience been so far?
So exactly because I'm finding it hard to find funding, I have been trying to connect with many different communities, right? And I think that currently in DeSci, there's more funding for people who want to find new ways of doing science than funding for actual research problems. I'm not sure I'm putting this correctly, but I still think right now there's still more money for discussing and for trying out models than money directly that could go into research, right? With notable exceptions. I know I am very curious about how DeSci funding for science projects are going to interface with universities, right? I am not committed to stay forever in a university, but while I'm in a university, there are two ways that I know that that projects can be funded. One of them is via gifts, where the people providing the gift have zero input on the project management or the IP. Okay? And there are sponsored research grants where funders, like not necessarily government agencies, but funders go give money for a project. They can ask for milestones to be completed, but universities, and in particular public universities, they will not cede IP. Everything that I produce is it belongs to the university that I am affiliated with, IP wise, right? The most generous thing that I've seen done is the university gives a first right of refusal, I think it's called, for things that come from the project, right? But I think that I don't fully understand how that mingles with the DeSci philosophy, right? I think it's going to be very easy to fund startups or institutes, but I do not see how DeSci can talk to universities, right? It's a big mess. This whole tech transfer IP stuff is a big mess. I don't understand how people giving money can be happy if they give money, but they cannot benefit from that in the future, right? So I think there are some kinks that need to be ironed out in this. I think there is one project that has been funded by DeSci means from the University of Washington, which actually is a public university, right, in the US. I think that's the only project that has been funded via this mechanism in the US. But I am very curious how this is going to be sorted out, because good or bad, universities are still producing a lot of the science, basic science. I mean, yeah.
Right, and that's the only quantum project funded by DeSci. Is that what you're saying?
What's that?
Only. Can you clarify which project was that? Was that the only quantum biology project?
No.
Was that
I don't think there is a.
Okay.
No. So it's something related to biosensors, I think, or something.
Okay.
It's nothing related to quantum biology, but it's the only project that I know in the US that has been funded by some sort of NFT.
Oh, okay.
IP NFT or something like this. I do not understand how this went through.
Yeah.
Exactly, because I don't understand how the IP got sorted out, especially by a public university, right? Because the people who gave the money for the IP, NFT, whatever, they probably wanted something in return. So I don't know how that got dealt with. I think it's a good first step. Right? And in any case, nobody's funding quantum bio. We had our own Bitcoin round. We didn't raise a lot of money. Again, because I think it's what we're doing is quite unique, and especially because we're developing instrumentation. It's very different from a, let's look at this molecular target. It's very different from a basic life sciences project. And also because I think that, I mean, the investment that we require is on par with investment needed for quantum experiments, which is usually, well, it takes more time and it's usually more expensive than regular biophysics experiments. So we're in a weird spot. And this is another reason why I would like to engage with your audience. If someone wants to fund quantum bio in a DeSci way, like, we should talk.
Yeah, no, definitely. And I think that's where it is now. I think I was maybe the DeSci round, that Bitcoin round, had a lot of diversity in different projects. But I think I'm not sure, there was very few academic projects in there, right? It was mostly just these sort of independent science projects, which I sort of moved into after grad school. So my background was I started a lab in my garage. I was still in grad school, and then ended up sort of selling the products of my research, turning it into a service, right? And then doing that for a couple of years.
Can I ask you a question? Like, how was that? Because it's so hard to do research outside academia for like weird reasons, right?
It was really hard. So I had to go through the process of tech transfer, where even though I had started the laboratory, did all of the research there. I had all the receipts for everything I did. All the equipment I bought is all there delivered to my home. Nothing to campus. Down to every little last detail. All the data I generated. I had a timestamp, date stamp, boom, this piece of equipment. To show that it was all done, off campus. It wasn't involving that, because I knew I would have to go through this process. And even still, it was like I submitted a stack of paperwork like this big after, I think I started working on it like 9 months before I finished my PhD. So it was 9 months worth of work, documentation. They were actually, the tech transfer office at UCSB was actually very, they worked with me a lot. They were very cooperative with that. They were, it wasn't too bad because I had all that documented. My advisor, the PhD advisor, said, he's been like, clearly, we don't have that sort of stuff. He's got it all down, because advisor, I guess, was more of the in-house expert, was using genomic technology. So that was, I guess, a different than most scientists' experience. They probably, have to go through more hoops or something, but I also didn't really have anything that was on the market yet. I had a provisional patent application and some preliminary data. And so that was about it, so it was, I guess, if I had a patent or something, if I had something that was more valuable, like if I had created a microscope, something like that, that would maybe be more difficult to transfer out. So yeah, that was kind of my experience. And once I did that, then even starting to do my own, basically start a business, right, off my research, right? That was difficult because, I didn't have the experience raising capital. It's the kind of, guess, a lot of scientists experience being an independent scientist. It's like running a business, which is a completely different skill set. So that's kind of, yeah, that's the fun of it.
That's very interesting. So here are my thoughts, right? So I think that this model might fit well to a certain class of experiments and not fit well to another class of experiments. For example, for quantum experiments, there is no way anyone can build a quantum experiment in a garage with like 200K, right?
No.
Those experiments, and alone. A quantum experiment is not something that you can build alone in the lab. Well, you can, but I mean, it's very, very hard. And you need like at least $1 million, if not more, right? So how do you start those things for this type of experiment, right?
Yeah, things that require newer, bigger machinery, bigger equipment, and especially building custom equipment, right? That's where a lot of the
it's not contemplated by the system. Yeah, there is no way to do this outside of regular academia for most cases.
Right. Unless you have a machine shop, and a glass shop, and electricity,
or a lot of money, or a lot of money,
or a lot of money, right? Yeah, you have to be somebody magical, or you have to have a lot of money. Yeah. So, but that's science and DeSci. There's not a lot of funding going around compared to traditional science, right? That Gitcoin round was, I think, it was fantastic to see all these projects, early stage projects get funding, but I think the matching round was about 500K. And what typical microscope, confocal microscope or something is going to be upwards of $1 million.
Oh yeah,
probably.
$1 million.
Exactly. Yeah.
Right. It's again I think that had I been an academic who could compete on equal footing with other academics, I wouldn't have turned to Gitcoin, right? I only started looking into those different sorts of funding mechanisms because the system is not ready for us, right? Because I sincerely do not think that we're getting treated in the same way. And actually, another experience, I won't give you the details because I'm not allowed, but I was part of a NSF judging panel. So the NSF has the National Science Foundation, that funds basic science in the US. They have different directorates, right? And those directorates, they assemble, say, twice, I think, a year to judge all the proposals that have been submitted, right? And I was called to be in the judging panels for one of those directorates. And what happens is that there's like 15 scientists, 3 or 4 are responsible, like mainly responsible for a couple of proposals. And you have, say, 12 minutes to discuss each proposal with the clock ticking down, right? Where people say things in favor or against. What I've personally seen was that for proposals that are highly interdisciplinary, like I mean, I didn't have a proposal for the directorate, of course, but for proposals that are very interdisciplinary, such as the proposal that I would submit, by minute 8 out of 12, you're still discussing the language, in the sense that people have not agreed on what they mean, what the proposal is really trying to accomplish, because I mean, it's so interdisciplinary. So I do not think that everyone says that, well, let's do interdisciplinary science, but I don't think interdisciplinary science is viable funding wise. And there are stories where interdisciplinary proposals were sent to like 2 different directorates, say at NSF, and then said proposal would have needed to be approved by both panels. So it's probability small p squared, right, that it gets accepted. So I think that governmental agencies still do not have sorted out how to
Mm-hmm.
best fund interdisciplinary science, which is
Yeah.
a pity, right? And which goes against everyone's like comments that, oh, interdisciplinary science is awesome and we like interdisciplinary science.
Mm-hmm.
No, we don't. Like, we silo people in departments, in universities, right? It's hard, and I understand. And in the end, people are. I really believe that people get a much better chance of being funded if they stick to their silos, right? If and actually also if they propose things that are slightly more incremental. I think that
Yeah.
funding mechanisms today favor incremental science.
Mm-hmm. Yeah, I mean, yeah, there's certain things, right? It's definitely, yeah, the traditional funding mechanisms. I
Yeah,
wholeheartedly agree,
traditional funding
right?
mechanisms. Yeah,
Yeah,
exactly.
yeah. But
Exactly.
then there's somebody like Elon Musk, who maybe he might be interested in quantum biology, right?
Elon, if you're listening to this, let's talk.
Yeah.
No, exactly, exactly. I
Yeah.
am. Oh, hi, Elon. Hi, people interested in space science. I mean, again, I really think that quantum biology,
Yeah.
or like my flavor of quantum biology, could be important for really space exploration. We need to
Yeah.
start thinking about
Yeah, for
other
sure.
things beyond, say, microgravity and cosmic radiation. We also need to start thinking about magnetic fields. Really, for sure, right?
Mm-hmm.
And yes, and again, and again, I think we have to think about like the private enterprise, right? So there are many discoveries in history that have been made by the private sector. A
Mm-hmm.
prime example is like Bell Labs, right? Bell Labs, again, you cannot compare the advances produced by Bell Labs to advances of any other universe, right? Bell Labs gave us a lot of new technologies. Um, Particle accelerators, they were first funded by private means. It was like using the money of
Oh,
a
wow.
guy who actually made money during the crash, the stock market crash of 29, and he made some like shady money, and then he wanted to give back, and he funded the first
Mm-hmm.
particle accelerators of the Lawrence Berkeley guy, I think. Yeah.
Oh, wow.
So there's a lot of
Yeah.
uh, or even the RNA vaccine pioneer. I forgot her
Mm-hmm.
name, but it's a scientist that was she's not American, but she do you know her name? Kariko
Mm.
something. I forgot her name.
No, I should though. Let me look it up.
So yeah, so her story is that she was developing RNA stuff very early on. She was effectively shunned from academia. She didn't get tenure. She didn't get money to run her lab. She had to leave
Mm-hmm.
academia. She went work for pharma companies. Uh, and nobody ever cared about her research for like 30 years. And now, since
Mm-hmm.
2020, because of the COVID pandemic, since then she has received the breakthrough prize. She has received like more than 50 prizes all of a
Oh,
sudden,
wow.
right?
Yeah.
So those are just some examples that I know of things that were developed in the private sector that probably couldn't have been developed in using regular funding mechanisms, right? So yes, I think there's a lot of room for the private sector for other models of science funding, and I really do think that nowadays boring research is way more likely to be funded than really cool stuff via usual means available to academics in particular.
Yeah. So DeSci is going to fund all the cool stuff. Is that what you're saying?
That's what I'm saying. Let's talk. That's exactly what
Awesome.
I'm saying.
Cool. Right on. No, and I agree. There's definitely the projects I see in the space now, and why I wanted to have you on. I think the research you're doing is super interesting. And you mentioned that there's having trouble finding funding for this, and it's just like, wow, like
Yeah.
that blows my mind. So that's anything else you wanted to end on? I think that's probably we've covered most of the stuff I wanted to talk about.
I want to say something. Can I say something?
Please.
About
Yeah.
quantum biology. So I think that for quantum biology in particular, there should be like an effort to do, as I've been trying to sell my vision, to do high-tech research. But I also think that there should be a effort by design to bring this field to the mainstream in a concerted way. I really think that actually not only for quantum biology, it's just that I'm in quantum biology, right? Academics and people involved in research should think of what's the best way to bring the research field that is at the edge to the mainstream, right? So things that I think about is, for example, 20 years ago, bioinformatics department didn't exist, right? So the question that I ask myself is, what should we be doing now, or what should we be thinking about now in terms of community building, curriculum development, so that in 25 years there are quantum biology departments in universities, right? So I would just like to maybe finish by saying that I encourage scientists in general to think a little bit about this, on how to do things by design, how to grow a field by design. That's something that I'm very interested in. Like, what makes a field mainstream? Like, why is it that string theory, for which there is absolutely no experimental proof, why you do not screw up with your career if you go into string theory, but you screw up with your career if you go into code fusion, or other fringe fields like this. Why is string theory? Why has it been brought to the mainstream, whereas code fusion wasn't? Right. So this is the type of thing that I think about. And again, one of my goals in my professional life is to bring this field by design to the mainstream, so that people, say in 25 years, in 10 years, can have an easier time than the time that I'm having now to run my experiments, to run my group, and to do good science.
Yeah, no, definitely. You'd think with science, people picture this, it's subjective, right? You picture this thing. It's but it's right, this kind of like pops, right? Pop culture of science, and it's just like these certain things get more attention, they get more funding. There's definitely cultural bias and publication bias type of things, so
totally.
We need to be more objective, and as
scientists need to be less dogmatic, right?
Yes.
I mean, it's crazy how scientists can be dogmatic, and it has to change. It has to change.
Yeah. Well, Clarice, this has been a wonderful conversation. Thank you for shedding some light on the small but big world of quantum biology, and hopefully all the potential future implications, down the road as we start, uncovering all the impacts on the world. So,
thank you so much for having me here, and I wish you all the success to this new podcast.
Thank you so much. All right. Well, that was another DeSci party, and we'll talk to you soon, everyone.